Good morning and welcome to Let's Talk Science. 2 years ago, our speaker, Dr. Jeffery Iliff, who's a professor of psychiatry and behavioral sciences and neurology, spoke in our series here and described the work that he and his colleagues were doing in revealing the functions of the glymphatic system, a brain-wide system that facilitates the clearance of waste products in the brain, and especially during sleep. They found that age and brain injury can result in impairment of the glymphatic system, and this may be one factor that contributes to Alzheimer's disease and other degenerative disorders of the brain. So much of this early research has been done on laboratory rodents. That's why there's mice in the title. But the next logical step, obviously, is research on humans. And Dr. Iliff is going to tell us about that and how difficult that prospect is. Please welcome Dr. Jeffery Iliff.
Thank you, Jack, for that great introduction. So I was here 2 years ago. One thing I thought about doing was just giving you the same talk and then having you raise your hand when you thought something was different. And then we could actually do a study, right? We could do a study about long-term memory. So what I'm going to be talking about today is really starting there and moving on. How many of you guys, when you follow the news, you see an article that says, new treatment for Alzheimer's discovered? So you see that about every week, right? How many of you are still waiting for that treatment to show up in your doctor's office? So what I'm going to be talking about is what I think is the reason for that separation, right? The fact that there's this gap between when we find a finding in the lab, often with rodents or very basic systems, and how do we start there and then get to the point where we say, no, this is a cause of Alzheimer's disease in people, that if we did something about it, we could cure or treat or prevent Alzheimer's disease, not in mice, because no one really cares about that except professors, but in people, right? So I'm going to be walking through really where that's, as a scientist, that's where I live. So I publish findings in mice and people say, aha, so we understand Alzheimer's disease, right? And the vain part of me says, well, yes, of course, we understand Alzheimer's disease. But the scientific part of me needs to say, no, we understand this in a mouse. We understand this in a human. And here's the gap between that and something that my mom or my dad or you or your friends or your children even at this point could actually make use of. So I'm going to be focusing on that gap through the lens of science. I'm going to be presenting a lot of data. I think that the public should see scientific data and not in cartoon form. I think it's my job to explain it so that you can understand it. And so if you don't understand my data, that was my fault, and I'm sorry for that in advance. That's not your fault, okay? But I do not believe in just showing cartoons to the public. I'm going to show you lots and lots of data. Some disclosures. I work in part for a little bit on my side time for a company that does try to make money off some of this. They pay me. I own a part of the company. You need to know that. So I have conflict of interest. I am presenting some data that company generated. The good news is I'm not trying to sell you their drugs or their devices, so that's good. I am a full-time employee of the U.S. federal government. So these opinions are my opinions and not the federal government's and the executive branch's opinions, and their opinions are not my opinions. I don't know why you're laughing. So 2 years ago, I talked about the glymphatic system. If this sounds like a fake word, it's because it is. It's a word that we made up 12 or 13 years ago. This is the biology that we've been describing for a little over a decade now about how the brain clears waste from inside, itself during sleep. Okay? I'm not going to redo that whole talk. Instead, I'm going to give you a cartoon, just like I just said I wouldn't. But I'm going to try to save some time. The glymphatic system, as we currently understand it, is that your brain is surrounded by, I guess there's folks over there, so I'm going to use my pointer so you can actually see, is surrounded by fluid, cerebral spinal fluid. For a long time, we thought that fluid was just a cushion, that the brain just sort of floated in it. But what we discovered was that fluid swashes into and through brain tissue along the outsides of blood vessels. So you have blood vessels that dive down through your brain. There's a space around those blood vessels called a perivascular space. That serves as a channel for fluid to go in. That fluid moves through the tissue. As it moves through, it picks up wastes and then eventually spills back out into that same fluid around the brain. And as that fluid washes through the brain tissue, that's the way that your brain gets rid of waste. Most of this biology has been defined in rodents. More recent work has actually been done in humans to make sure that what's happening in a mouse and a rat is also happening in a person, right? But some things we know about this. One is this process is much faster than we thought it was. It's very fast. It's much more anatomically organized, meaning your brain uses these blood vessels as a scaffold, as an organizer of that fluid flow. We know that there are drivers of this process. We know that the pulsing of your blood vessels is one of the driving processes for this. We know that the neurons inside your brain during certain phases of sleep, they pulse synchronously. Your neurons do that during what's called slow wave sleep. That's a driving force for this process. We know that the support cells of the brain, which have these water channels that surround the blood vessels, that water transport through those cells is an important regulator of that process. And we know that this biology contributes to the clearance of things. I'm going to name 3 things in particular: amyloid, tau, and synuclein, alpha-synuclein. Who's heard of amyloid? Right? Amyloid's one of the things that builds up in Alzheimer's disease. Who's heard of tau? Tau is the other thing that builds up in Alzheimer's disease. Who's heard of alpha-synuclein? Less. That's the thing that builds up in Parkinson's disease and Lewy body dementia. Okay? So these are the proteins that aggregate and stick together in the brains of people who are developing Alzheimer's and Parkinson's. So those things are cleared by this biology. This will be an interesting exercise. So when I give talks to graduate students and undergraduate college students, I flash one of these up and everyone pulls out their iPhone and they take a picture of the QR code. I was sort of debating whether to take these all out of this talk, but I didn't want to make any preconceived judgments about this particular audience. So a couple of things. This is a process that's more rapid in the sleeping brain that we observed. So this is a feature of sleep. This contributes to the clearance of amyloid and tau and synuclein. Okay? These 2 things have led to 2 general assumptions or beliefs. Note that I'm using the word belief and not fact. These are things that we think. These are not things that we know. One is that this sleep active clearance of these proteins is one of the reasons why sleep restores cognitive function. And the disruption of sleep erodes cognitive function. We think that linkage is true. We also think that this role in connecting it to amyloid and tau clearance is one of the reasons why in epidemiological studies, so studies of large groups of people, we see that people who sleep poorly have higher rates of dementia. That's true. People who sleep poorly have higher rates of Alzheimer's disease. That's true. We think that a failure of this biology is the reason why those 2 things are linked. That this is the thing that's going wrong that links bad sleep with dementia. Those are beliefs. These are cartoons. No one can lie about something quite so much as a scientist talking about their own work. You should know that. So if you don't, you shouldn't take my word for it. If you're a person who reads articles and likes to read the scientific literature, This QR code, if you scan it with your phone, and some others that are coming later, will take you to lists of articles that give the evidence supporting these claims that I'm making. If you don't want to do that, just take my word for it. Oh, I'm so sorry, Jack. Here, I'll put it back up for you. Okay, here we go. So if we think there's a connection between this biology and its failure and the development of Alzheimer's disease, why do we think that? Do we have any evidence for that? And here I'm going to make a really important distinction. This is a distinction between preclinical, meaning before the clinic, rodent work, right? So a lot of times preclinical is a synonym for mice and rats, and clinical work, which is work in humans, right? We get data from both types of studies, and both types of studies allow us to ask different types of questions. They don't answer the same questions. So we're going to talk first about what is the preclinical, mice and rats, evidence that glymphatic impairment contributes to Alzheimer's disease. What's that tell us? So a little bit about my lens as I think about Alzheimer's disease. I'm not a clinician. My parents still hope that one day I will be a real doctor and not just the person with a PhD. I don't know when they're going to figure out that that's never going to happen. So when I think about Alzheimer's disease and Parkinson's, I think about it biologically. I don't so much think about it clinically. I think about these diseases as diseases of protein misaggregation. So proteins that are sticking together when they shouldn't. In the case of Alzheimer's disease, we already talked about it, amyloid plaques in the spaces outside the cells, tau tangles in the spaces inside the neurons. These are the two things. If you don't have these, you don't have Alzheimer's disease, right? If you do have these, nowadays, even if you don't have cognitive impairment, you still have Alzheimer's disease. It just hasn't shown itself yet, okay? So these are the pathological features of Alzheimer's disease. So these are diseases associated with protein aggregation in the brain. We know that across these different diseases, like Alzheimer's, like Parkinson's, like Lewy body dementia, they're all influenced by different non-genetic risk factors. So there's genetic drivers for sure, but then there's some other stuff that make you more likely to get Alzheimer's disease. Aging. You don't even think about that as a risk factor, right? It's not really a disease, but it's a thing that happens to you that makes your chances of getting Alzheimer's disease higher. Chronic comorbidities like hypertension and diabetes. Cerebrovascular disease, right? So strokes, small vessel disease, those kinds of things. Traumatic brain injury increases your risk of dementia. Even concussions, right? Even mild traumatic brain injuries. Chronic sleep disruption, this is new. Chronic sleep disruption has emerged as a risk factor for Alzheimer's disease, okay? So there's something about the aging, the injured, or the sleepless brain that makes it vulnerable to this process of protein misaggregation, okay? That's my lens. If I want to answer this question of does glymphatic function and its impairment contribute to the development of these diseases, there's a set of questions I can answer in a mouse, right? So one is, well, does this biology actually clear this stuff away? Because if it doesn't clear this stuff away, it probably doesn't have much to do with Alzheimer's disease, okay? Second question I can ask is, if I take an old mouse, if I take an injured mouse, and if I take a sleepless mouse, do I see impaired clearance? If I do, then that supports the idea that that failure of clearance is why aging, brain injury, and sleep disruption make Alzheimer's more likely. It could be the link, right? If it doesn't, then it probably isn't the link. I can answer that in a mouse. I can also do things in a mouse that I could never do. What I can't do in you is say, what I'm going to do is I'm going to disable your brain clearance for about 10 years and I'm going to see how it goes, right? And we think it's going to be bad, so get ready. We can't do that, right? But we can do that in a mouse. And it doesn't take us 10 years. It only takes us a couple of months because mice get old faster. So I can ask the question of, if I impair brain clearance, do I get more plaques? Do I get more tangles? What that tells me is it is sufficient. It is able to drive pathology. Pop quiz. If you're able, everyone raise your hand. if you're able. Put your hand down if the answer to this is yes. I impair lymphatic function. It enhances pathology. That does not prove that it causes Alzheimer's disease. Put your hand down. I know that was a very confusing instruction. I just made that up on the spot, and so that was very confusing. The point is, just because I can make pathology in a mouse doesn't mean that's actually what's happening in a person. Mice and people are super different. In fact, lots and lots and lots of studies show that I can prevent plaques and tangles in mice, and they don't work at all in people. And that's true in Alzheimer's research. That's true in stroke research. That's true in traumatic brain injury research. It's a massive failing of what's called translational neuroscience, is that the things that we discover in rodents often, not always, don't work in humans. Now, that doesn't mean that we shouldn't do mouse research, but it means that we need to be careful in interpreting mouse research to understand what are the false positives, what are the false negatives, right? There are real success stories. There's a whole new class of migraine drugs called anti-CGRP blockers, right? So these are things that block a certain molecule in the brain. They totally work. They revolutionized migraine treatments. All of it came out of mice. Totally worked, right? So there's examples of mouse work that does work, but there's a whole bunch that doesn't. So the job is to figure out what works and what doesn't, what translates, what doesn't. Okay. So I'm not going to go through all of these and all of this work. We could just do that for a week. I could just come in here and talk for 8 hours a day for a week. And if you don't have dementia now, you're going to have dementia by the time we're done with it, right? So instead, I'm going to give you 3 short vignettes touching on each of these pieces, okay? So first question, does this brain clearance system contribute to the clearance of amyloid and tau? The answer that we discovered in mouse is yes. If I inject amyloid or tau, I'm showing you amyloid, into the brain of a mouse, first thing I see, here it's labeled in green, is it moves through the brain of a mouse. This is a blood vessel. There it is. That's another blood vessel. There it is. It moves through the brain of animals along blood vessels. So it is using these pathways. Second, I happen to have a mouse that I didn't do it, someone else did it. Someone else genetically engineered in such a way that it doesn't do brainwashing. This is a mouse that can't wash its brain. If I inject amyloid into that mouse, in a normal mouse, it clears about 70% of the amyloid in the first hour. That's how the brain normally functions. If I kill that brainwashing, the clearance is slower, right? So what that tells us is that this washing system, it's occurring along these blood vessels, right? And the clearance of amyloid and also tau and also synuclein are cleared by this process, okay? Second, so check. Is lymphatic function impaired in animal models of non-genetic AD risk factors? So things that make you more likely to get Alzheimer's disease. Do we see impaired washing there? This is aging. I'm not showing you other things. I'm just showing you aging. The only thing wrong with these animals is that they're old. And that's not really wrong. They're just old animals. These are healthy young mice, 2 to 3 months old. That's like 18 to 20 years old, right? So they're in unwise relationships. They have high insurance premiums, and they're making bad decisions. And these are 18-month-old animals. This isn't old. Mice live to about 2 years, so 18 months old, right? They're starting to think about contributing to the IRAs and things like that. So these are old mice, but not so old mice. So if I measure the movement of fluid into brain tissue, I see the healthy young mice, there's a bunch. In old mice, there's much less. If I measure clearance out of the brain, we see the same thing. So I inject in amyloid. This is how much got cleared in 1 hour. So in a healthy young mouse, 70% is cleared in 1 hour. 12-month, 18-month-old animals. As animals get older, they clear amyloid slower and slower and slower. So this clearance is impaired in aging. And other models showed that it's impaired in mice with diabetes, in mice with hypertension, in mice with small vessel disease, in mice with traumatic brain injuries, in mice with strokes, in mice that when you disrupt their sleep acutely, in mice where you disrupt their sleep chronically, right? So in all of those cases, we see impairment in those settings. So check. Third, if I do the experiment where I stop all of your brain clearance for 10 years, what happens? That's what we're doing in a mouse, right? So I've got mice, right? We do some mouse breeding. We make some arranged marriages. We get some baby mice. It works out okay. Some of those mice, these mice are genetically engineered to make all kinds of plaques. That's what this red is amyloid plaques, just like people get. The mice that have a functioning system, they get a certain number of plaques. That's black. In the mice that have a disabled system, you can even just look, more red, right? More plaques with less washing. So if I disable brain washing, I get more plaques. That's true of amyloid. That's what I'm showing you. What I'm not showing you because of time is same thing with tau, same thing with alpha-synuclein. All of those things are driven by this. So does impairment contribute, drive pathology? The answer is yes. I showed you 3 pieces of data. I made some pretty big claims. Those claims are supported by probably 30 different articles. If you want to go check out those articles, if you got a lot of time on your hands, and if you're kind of nerdy, this is where you can go to this list. It's a different list. I don't think it's a nerdy list. I think it's a great list. But what about humans? Who cares about mice? What do we know about humans? So we're going to get a little bit technical here. Have any of you guys ever heard of a journal club? Who's heard of a journal club? So a journal club is something, my parents didn't go to college. I went to college. I had a girlfriend in college who was doing research, and she told me about a journal club, and I was like, what's a journal club? And she described it to me, and I said, only the nerdiest people on earth would ever do a journal club. What a journal club is when scientists get together and talk through a new paper, right? They call it a club because this is their idea of social interaction, right? When you're in graduate school, sometimes it involves pizza and beer, right? That's how it is. So that's journal club. So we're going to have a journal club. I don't know about the pizza and beer. Maybe that'll arrive later. We're going to talk over 4 papers. These are the papers that established the role of sleep in this brain clearance in mice. Okay? We're going to walk through 4 key sets of findings. And then we're going to ask the question of how are those things holding up in humans? So we're making a direct mouse to human comparison in our journal club. A lot of data. 2 things I want you to pay attention to on this page. First, these are 2 different papers that we published, 2012, 2013. Those were great years. That was the very start of this whole story. 2 things we discovered studying mice and rats. One, we're using a kind of imaging called 2-photon microscopy. Not an important detail. The important thing about this is it allows us to image into the brain of a living mouse. What we did is we injected a tracer into the fluid that surrounds the brain. We then imaged the mouse over time. And what we observed, this is the surface of the brain, was that tracer went along the surface of the brain along, what's this thing? It's a blood vessel. So it went along the surface of the brain, along the outsides of blood vessels. You can see right here, there's a blood vessel on the surface of the brain. You can see the tracer around it in the perivascular space. Where that blood vessel turns and dives down into the brain, that's what this is. This is the cross-section of a vessel diving into the brain. What do I see? I see a moon-shaped space around it that is filled with fluid. That is the fluid following the blood vessel down into the brain tissue. This is the data that told us that this circulation of fluid is perivascular. It is organized around the blood vessels. Second, MRI. 2-photon imaging gives us a view that's about this big. Like, can't even see it. It's such a tiny view. We can only see a tiny fraction of the brain with 2-photon microscopy. MRI, most of you have probably had an MRI at some point in your life. Some of you have probably had MRIs on your brain. MRI of your brain is 3-dimensional. It's intrinsically 3-dimensional. So you can see the whole brain. That's where the value is. One of the things that we see with MRI is this is something we inject in an MRI contrast agent into the CSF. We see that movement is happening everywhere. It's not just happening in this tiny spot where we're looking. It's happening through the entire brain. Brain-wide organized transport. Okay? What about people? It turns out you can do the same thing in a human that we did in a mouse. So I can take a human, I can inject MRI contrast agent into their CSF at their lumbar spine, kind of where you would get an epidural, and I can image where that contrast agent goes over time. Now, it's called an off-label use of the contrast agent, which means the FDA has not approved that use of that contrast agent. So it's off-label, but the good news is there is a lab in Oslo, Norway, where they think this is a good idea. And so they've been doing this in people ever since 2015. And this is the group that has validated much of this biology in people using this technique. So going to here, what they did is they inject it. First, they take a scan of the person before the contrast. That's over here. No contrast. This is just the brain. Then they inject the contrast and they image the person over time. An hour later, 3 hours later, 4.5 hours later, 24 hours later. Okay? Couple things that you see. One, first, the contrast enters the CSF spaces around the brain. You see early enhancement, so the contrast starting to move into brain tissue, right? And then the next day, what do you see? You see that the whole brain is brighter. That's because contrast agent has moved through the entire brain, right? That's showing that this exchange is happening through the entire brain in people. They recently, so this is 1.5 years ago, they published a paper where they used high resolution MRI. So they used some fancy stuff. And what they showed was that this movement, if you look, what do I see? I see a blood vessel that is surrounded by contrast. I see a blood vessel that's surrounded by contrast. This movement is in fact organized around the blood vessels, just like we saw in rodents. So at least the organization and the extent of it is the same in people as it is in mice. Good news. What about the sleep thing? Is it faster in sleep? What's that about? So this was a study, this was published in the journal Science back in 2013. This was the one that made us all famous, right? So the study was inject that tracer into the fluid that surrounds the brain of a mouse, let the mouse be awake, orange, let the mouse be asleep, green. What do we see? We see a huge difference. When the mice are asleep, the exchange is fast. When the mice are awake, the exchange turns off, okay? If we flip it, and if we inject amyloid or some other molecule into the brain and we measure how fast does the brain get rid of it, we see that when the mouse is awake, they get rid of it at a certain rate. When the mouse is awake, the rate of clearance is slower. It's shallower, so they get rid of it less fast, almost twice as slow. So the rate of clearance in mice, in the waking brain compared to the sleeping brain, is twice as slow in mice. What about people? Yes, sir. So we also studied a certain anesthetic agent, ketamine and xylazine. It turns out to be really hard to study sleeping mice. And so we study a type of anesthetic that mimics many of the features of deep sleep. So that's ketamine, xylazine. Good catch. Interestingly, this was 2013, right? So in August of 2013, that paper was published. In August of 2013, all of the media said, sleep, your brain clears itself when it sleeps, right? Many of you probably read that. How long was it before that was validated, shown again, either in another lab, because maybe they screwed it up, maybe they were lying, right? Or it was shown in another species, like a human. How long was it? Everyone, put your hand up if you can. Put your hand down when you think you get to the time. Oh, you guys aren't participating. No one wants to put up your hand? Okay. Put it down when you think you get to the right time. It was validated in 1 year. Put your hand down. It was validated in 2 years. Put your hand down. It was validated in 3 years. Put your hand down. So 3 years. If it was validated in 5 years, put your hand down. So 5 years. It was validated in 7 years. Put your hand down. All of you are wrong. It took 8 years to validate it. 8 years that we were saying, and by we, I mean the media, scientists, people were saying, the brain cleans itself when it sleeps. We said that for 8 years before a second lab showed the same thing, and before it was even shown to be true in humans. It's a good thing that it turned out to be true. Otherwise, we would have been talking nonsense for 8 years. So we're back in Oslo, right? So they do crazy things in Oslo. It's a very simple study design. The study design is this. They take participants. They randomize the participants into two groups. So they separate it into two groups. Red group, blue group. Both groups, they inject the contrast agent in the afternoon. Both groups go in for scans in the evening. And what they do is they measure how much of the contrast went into the brain at that evening time point. And what they see is that there's equal loading between the two groups. So both groups are the same in terms of what is in. And then what happens is blue group gets to sleep overnight. Red group doesn't. Red group is kept up all night. And then they come back the next day and they scan how much of that contrast has cleared since the night before. And what they observe is that in the sleeping group, here I'm going to use my highlighter, blue sleeping group cleared a certain amount. Red sleep-deprived group cleared less. What does this show? This shows very clearly that sleep deprivation slows the clearance of at least gadolinium, this contrast agent, out of the brain. Clearance is sleep sensitive. Both groups were allowed to sleep the second night. So both groups are sleeping. That deficit remained even after the second night. So they weren't able to catch up even with one night of sleep after. Okay. This was the first validation of that finding initially in rodents. In humans, it took 8 years. Okay. That's one of the big reasons why from the time that you discover something to the time that you can actually do something in a person's life about it, it took us 8 years to figure out how to answer this question in a person. Because you can do almost anything within certain ethical boundaries to a mouse, and there's not a lot we can do with humans, right? I can't do some of these studies into people, and it's very technically challenging. So let's talk about sleep. What is it about sleep that is driving these processes? And here we're going to get even more technical if you guys are ready for it. This was a very neat study. So this actually goes back to the prior question. So one of the things that some of us noticed when we were doing rodent experiments early on was that depending on what anesthesia I use for the mouse, I got different results. So sometimes we use inhaled anesthetic on mice. Sometimes we use injectable anesthetics. There's a bunch of different anesthetics you can use. And one of the things that we saw was that there were some anesthetics that we used that shut everything off. And there were other anesthetics that we used which turned everything on, right? Which is bad for an experimenter, but it turns out to be useful in giving us some information. So there was a person, her name was Lauren Hablitz. She was in the lab after I left the lab in New York. She did an experiment it must have been 1,000 mice. She used like 1,000 mice for this. It was such a huge study where she used 1, 2, 3, 4, 5, 6 different types of anesthesia, right? It's not important what these are. So this is an inhaled anesthetic. These are injected anesthetics, right? This is a combination. One of the things that she saw was that some anesthetics like this one turn the system on, faster clearance. Some of the anesthetics turn it off, slower clearance. Good to know. One of the other things that she did is she measured brain activity in all of those mice. And when you measure brain activity using a technique called EEG, electroencephalography, right? So these are electrodes all over the brain that are measuring all the neurons firing. Sometimes neurons fire fast and all on their own. Sometimes they fire slow and all together. Okay? So fast and asynchronous, slow and synchronous, right? 2 types of firing. One of the things she was able to do was look at the brain activity across all of these groups and figure out how does it relate to brain clearance. What she saw was this is high frequency activity, so higher frequency or more high frequency. With more and more and more high frequency activity, you got less and less and less and less clearance. So the more active and jumpy the brain was, the less of it there was, the less activity there was. This is low frequency, synchronous activity. This is slow, heavy activity, right? The more of that there was, the more activity there was. So this study told us that slow wave activity, that's slow wave sleep, slow wave activity actually drives this process. And high frequency activity, which is characteristic of waking, suppresses it. interesting point. Guess what the news article from this paper said? This news article said, the brain cleans itself during slow wave sleep, which is your deep sleep. The brain cleans itself during slow wave sleep, okay? Does anyone see something wrong with that statement? The brain cleans itself between slow wave sleep. The experiment was evaluating 6 different anesthesias. Were any of these animals asleep? No, these animals were anesthetized. Sleep and anesthesia are not the same. They're not. And different anesthesias create different electrical activities, right? And so what this tells us, it doesn't tell us slow wave sleep drives this function. It tells us that if I give you a drug that promotes slow synchronous activity, it does it. They didn't study them in this experiment. They didn't do it. So, and this is the trick. It makes sense that the drug that looks most like slow wave sleep, that's KX, it does increase this process, right? But it's not the same. Mice don't even have, like we call slow wave sleep N3 sleep. Mice don't even have N3 sleep. We don't segregate mice sleep that way. Their sleep isn't like our sleep in that way. So that's an example of the story getting out a little bit ahead of what the data actually said. Is that okay? It depends. Is your life enriched by knowing the difference between mouse and human non-REM sleep? Not really. Is that an important distinction? I don't know. Maybe, maybe not. Who knows? But it's important to think about that sometimes what you read in the press or what even scientists say might go further than what the data says. Important to know. And that sounds almost political. That's just true. And that's true through all the sciences. We're all like this, right? Because we're all telling stories. Reduced heart rate. This is interesting. The slower the heart rate, go this way, the slower the heart rate, the more activity. So a slow beating heart, faster activity. Interesting. Here's another one. This is kind of a technical piece. This is an example of electrophysiology. So we stuck electrodes into the brains of the mouse. The electrodes there's 2 electrodes placed very close to each other. So they're only about 100 microns, so 0.1 millimeter apart from each other. And what happens is you release a molecule from one of them and you measure it at the other one. And so what you're measuring is how does that molecule pass between them? And one of the things that we know is that as the space between these electrodes changes, how this travels changes. So if that space is big and wide open, it travels very quickly. If that space is very small and tangled and tortuous, it travels through a much harder route. And so we can use this approach to measure how big is that space? How confused or convoluted is that space? And one of the things that we saw was that this extracellular volume, how big the space between the brain cells is, actually got way bigger, like 50% bigger during sleep. So one of the things that's happening during sleep is that the spaces between the brain cells are actually widening out. If you think about a freeway, if I keep the number of cars the same and I double the number of lanes, what happens to the speed of traffic and the sort of the general experience of people in the cars? It flows faster and everyone's happier, right? Well, if I don't change the number of cars, that is true, right? Everything moves more easily, right? And if I make that super small, if I take I-405 and I bring it down to a single lane, and I don't reduce the number of cars in some way, everyone hates their lives, right? Same thing with the movement of fluid and solutes through tissue. Bigger is faster when you're talking about transport. So the brain's space in between its cells is actually getting bigger and smaller. Okay, so this is the new data. This was published this last summer. It's going to get a little crazy here, so get ready. The trick is, how do we measure this stuff in people, right? And it's hard for me to tell you about measuring this stuff in people without getting a little bit technical, because it turns out you have to use sophisticated approaches to do this. But I'm going to try to explain it in a reasonable way. This is a collaboration between a couple of really great collaborators. We're using two different measures that are important. One is we're using MRI to measure how much fluid and solute is going into the brain and how much is leaving. So we're measuring inflow in and we're measuring clearance out. So we can measure both. We're also using a new technique. This is a new thing. A device that has in-ear electrodes. So a person wears it. You put the electrodes in your ears, and it passes current between those electrodes. So electricity is literally going through your brain between these currents. But the currents are very small, and we're told they're very harmless, right? They are. They're supposedly harmless. So just a little bit of current is passing between them at high frequencies. So when the current is going really fast, it's able to go through the brain sort of where the cells and everything in the brain is kind of invisible to it. It just passes straight through. But as the current pulses slower and slower and slower, what happens is the cells of the brain start to create obstacles to the movement of that electricity. And the electricity has to start to wind its way in between the cells. And so we can use how that current moves between those as a way to measure how big is the space between the cells in people. So this is a new use of this technique in people for the first time. This is the first time it's ever been described. We did a study. Part of the study was carried out in Florida. Part of it was carried out right here in Seattle at the UW Medical Center. These are older people, 50 to 65 years of age, cognitively intact. So these aren't people with dementia or cognitive impairment. We had what's called a crossover design. So participants came in twice. One time when they came in, they slept normally overnight. The other time they came in, they were kept up all night. Okay? So sleep deprivation. The next day, they were awake for a while. The sleepers stayed awake, and the sleep-deprived people got to try to sleep. Okay? So it's a complicated design, but the idea is we have data from each person sleeping, sleep depriving, waking, and undergoing what's called recovery sleep. So 4 different sleep conditions in each person. So it's a very powerful technique because we can see, because there's variability between us, but if I do the same thing again and again and again and again, there's much less variability between me and myself, right? So with this design, I can get, and then we did an MRI here starting in the morning into the afternoon. And so what it allows us to do is understand, how does the sleep that happened before influence how the contrast goes into the brain? And how does the sleep that happens later affect the contrast clearance out of the brain? So with that design, we can actually look at the movement of stuff in, and we can look at the movement of stuff out. And we're measuring all their brain stuff in between, okay? That's a lot. So one piece of data, and then I'm going to show you a table that's not going to make much sense, but there you go. It's the best way I've thought of to show this data, unfortunately, is in a table form. And sometimes data is like that. It just isn't good looking. So first, this is measuring the brain's resistance to fluid flow, right? So that's how wide is the space between the cells. As resistance goes down, there's more space between, easier flow. So this is in people who are sleeping, we see that resistance declines as they're sleeping. In the same people, when they're being sleep deprived, no change. Okay? So this was the first confirmation. Here we are. How long did it take after the publication of the first? So the finding in mice was the space gets bigger and smaller with sleep and wake, right? What year was that again? 2013. What year is this? 12 years to replicate that finding in another lab and in people. Because it's hard, okay? So what we saw is during sleep, the resistance changes, more space between the cells. Even when these people were awake, they were only awake for a little while. What happens? Resistance starts going up. So the spaces start closing off. These people are sleeping just a little bit. What do we see? The resistance starts going down, right? So this is the very first confirmation that the spaces between the brain cells are actually changing in people sleep. Interesting. One of the things that we can then do is we can ask questions so that high frequency relationship that I talked about in humans where there's more washing with less high frequency. So high frequency is bad. There's more washing with low frequency. Low frequency is good. Is that true in a human? The answer is yes. So in this case, green is good, red is bad. We're just going to color code it really easy. These are the outputs of complicated statistical models, right? So we're trying to use color coding. So slow wave activity, right? More of it, you get more wash in, you get more clearance. Slow wave activity, that is human slow wave sleep. High frequency activity, the more of it you get, the less in you get. No association there. Heart rate, remember heart rate. As the heart rate got slower, things went faster. Well, as the heart rate, here you have to kind of flip it. As the heart rate gets higher, this gets lower. So if you turn that on its head, it's true. Lower heart rate, more wash in, more wash out. What about resistance? That resistance turns out to be probably the most important factor. It turns out that the resistance of the brain is the valve for this whole process. Okay. So what do we know about people? We know in people, this clearing does happen in people. It is organized around the axis of the brain blood vessels. So that organization is true. It is brain-wide and it is faster than you would expect if this wasn't some sort of facilitated rapid process. It is sleep sensitive. It is sleep dependent, right? So this is a process of sleep and it's associated with low frequency activity, slow wave sleep, lower heart rate, which probably connects with parasympathetic activity, which some of you have probably heard of, and changes in the brain's resistance to fluid flow. Okay, last bit. What about human disease, human Alzheimer's disease? So remember this framework. Alzheimer's disease is a disease of protein misaggregation influenced by these different factors. Remember there were 3 questions we could answer in rodents? There's 5 questions we can answer in humans. And for the next 4 hours, we're going to talk through those 5. That's not true. First, I can take post-mortem human brains, right? Which we have a brain bank at the University of Washington that people donate their brains with when they pass away. And we use those to study exactly these processes. I can look in those brains and I can say, hey, do I see the same thing in people with Alzheimer's disease and people with these risk factors that in a mouse impairs this process? I can ask that question in people. Second, if this is a thing that's driving Alzheimer's disease, then I would expect that the genes so genes make proteins or they describe how to make a protein, right? So I can describe whether gene variants, so naturally occurring variation in human genes that are connected to this biology, they should confer risk for Alzheimer's disease and they should confer protection from Alzheimer's disease. So I can look in human genetic studies for evidence of this. Third, I can look at the question of, hey, I can measure amyloid and tau clearance from people. Does this process clear amyloid and tau in humans? Fourth, I can measure this process in humans and I can ask, hey, is brain clearance, now that we can measure it, is it slower in old people? Is it slower with the hypertension and diabetes? Is it slower in people with small vessel disease? Is it slower in people with sleep apnea and insomnia? I can answer that question now. And actually now that we're doing this, we have 8 different ongoing clinical studies in humans answering that question. So we're studying this biology in aging and Alzheimer's disease, obstructive sleep apnea, REM sleep behavior disorder, which is believed to be connected to Parkinson's disease and Lewy body dementia, blast TBI, so in Iraq and Afghanistan veterans. We're studying this actively now. And last, this is the smoking gun. If I change glymphatic function in you and in me, Do I change the development of Alzheimer's pathology? Can I actually change the course of the pathology itself? Can I prevent the onset of cognitive impairment? Can I prevent clinical decline by changing lymphatic function in people, right? That's smoking gun type of evidence, okay? So what we're gonna do is we're gonna assess, I've got like 50 slides that I'd be glad to go through in my 6th hour of this talk. But what I'm gonna do is I'm gonna sort of walk through what we got. Do we see changes in human postmortem brains that in mice are consistent with an impairment of this process? Yes. We've seen that in probably a dozen different studies. That's absolutely true. Do we see that gene variants associated with brain clearance are risk variants and protective variants for Alzheimer's disease, right? So if I have different gene mutations, am I at higher or lower risk? The answer to that is yes. Does lymphatic function contribute to the clearance of amyloid and tau in the human brain? Remember, what year was the first evidence of amyloid clearance through this process in mice? 2012. How long ago was that? 14 years. The very first paper demonstrating this, we're going to publish it next month. 14 years it took for us to get evidence that the human brain clearance process contributes to amyloid and tau clearance in humans. It took us 14 years. How about this? Is lymphatic function impaired in Alzheimer's in the setting of these risk factors? We don't know. We're starting to study it right now. So I just described some studies that we're doing. There are now groups all over the world who are now chasing these questions, right? So in Europe, in Asia, in Australia, here, not so much in Africa or South America yet, but maybe soon, they're studying these questions. We don't know yet. What about this? If I modulate lymphatic function, do I impair amyloid and tau clearance in humans? To be honest, if I had an answer to this question, I wouldn't be here right now. I'd be on a yacht in the Mediterranean Sea with my family, celebrating the Christmas. No, this is the thing, right? Here's something interesting. Now that we can measure lymphatic function, I can tell you that a group I'm a part of is going to publish probably this spring the first evidence in humans of a drug that enhances brain clearance in people. And it enhances amyloid and tau clearance in people. Does that mean it will prevent Alzheimer's disease? No, right? We don't know. We don't know the answer to that. But it is the first step toward this process. It will be the first study that shows that in humans, you can change lymphatic function and change amyloid and tau clearance, okay? That's it. That's where we're at. So does lymphatic function cause Alzheimer's disease? We don't know. There's several pharmacological approaches in rodents that seem to modulate lymphatic function, but we're just starting to test those in people now. It looks like there's a bunch of corroborating data in humans that suggests that targeting this might be helpful, but we haven't done this study. This is actually a hard study to do because we have to start giving people the drug and then wait 5 years or wait 10 years to see what happens, right? That's a hard process. So there's strong data, preclinical rodents, quite a bit of supporting clinical data supporting the idea, but we don't know yet. So that's the end. That's the story, to be continued. It's a story that started back in 2012. It's a story I told you about 2 years ago, and this is where we're at, trying to understand, okay, we're moving, but you'll notice the waiting times for some of these steps, 14 years from an initial observation in a mouse to being able to answer that question in a human, right? So you can imagine we still might be 5, even 10 years away from leveraging this biology to actually changing how human diseases are treated. So I went a little over time, sorry about that. Glad to take questions.
This is incredible. Stay tuned. We're going to have questions. We'll take it by sections, and let's start over here. There's a
You talk.
Go ahead. It's on.
You talk about sleep deprivation, and can you give any sense to those of us seniors who struggle with sleep? some measure of what's acceptable and what isn't?
So the question is, how much sleep is okay? And to be honest, I don't think there's a good answer to that. So the American Academy of Sleep Medicine says 7 to 9 hours. How many of you all get 7 to 9 hours of awesome sleep every single night? Every night? Good job. So that's not many. And I'm finding I'm 48. It's getting harder and harder and harder. What we don't know is whether that changes as you age. What we don't know is if I get 8 hours of sleep, but it's not that great, right? Versus 6 hours, that's awesome. We don't have that kind of granular understanding. What we do know is that just epidemiologically, if you sleep less than 6 hours, you have about a 50% higher incidence of dementia. If you sleep more than 9 hours, you also have a higher risk. And their thinking is because your sleep is so crappy, that's why you're sleeping so much, right? Or you have some other condition like depression or some other things like that go with sleep disruption, right? So we don't know the answer to that. And it could very well be that it's what is in your sleep itself. So one of the things, if we took all of you and did a sleep study, what we would find is many, in fact, maybe most of you don't even have hardly any slow wave sleep at all. That's a thing that we know goes away as you get older. You lose it. And is that bad? We actually don't know the answer to that yet. And it's actually hard to understand the answer to that because it's hard to take a person who doesn't have slow wave sleep and just give it back to you and see what happens, right? And so I don't think we have a good answer to that. I think focusing on understanding what gives you good and restorative sleep. So I track my sleep because I'm not an anxious person, and I find that helps me understand cause and effect. So I know if I have 2 beers, I don't sleep well that night. If I have 1 beer, I sleep fine. If I have no beers and go for a run early in the day, I sleep even better. I know that because I paid attention to what my watch tells me, right? So understanding that for yourself, I think, is important.
And my question is over here, have you done any of these studies against heavy meditators?
Oh, so meditation is a great question. We have a study that is wrapping up right now looking at Tai Chi. Not meditation, not the same, but it is a martial art that includes elements of mindfulness, elements of respiration, elements of body posture, elements of sympathetic and parasympathetic tone. And so we actually have a total of 30 participants. We're finishing our last couple of participants now. We suspect it will enhance this process, but it's hard to say. This is the hard part. Is it the meditation that does it? Or is it the type of person who meditates that has the other things that cause this? You laugh, but understanding what so if I take a person who would never choose to meditate and make them meditate, are they going to get the benefit? Or do I have to convert them into the type of person who would want to meditate? I don't know.
I take a couple medications at night when I go to bed, and they say make you drowsy. So does that affect the quality of sleep? And also people that take a sleeping pill,
Yep.
Does that affect the quality?
So many medications affect sleep. Many medications that people take for sleep create sleep that is maybe not the sleep that you want to get. So a lot of people take antihistamines. So like NyQuil has an antihistamine in it, right? Or Benadryl. People take that for sleep, especially older people, because that's a thing that they told you to take when you were pregnant or whenever, right? It turns out that antihistamine use for sleep is actually associated with an increased risk of dementia. Ambien, which is commonly used, produces sleep that's real light on REM sleep. Trazodone, which is commonly used for sleep, even though it's an antidepressive, produces a different type of sleep profile, right? So different drugs produce different types of electrophysiological profiles. It's very important that if you're taking medications for sleep, that you're running that by your PCP, and that PCP has good current experience thinking about sleep issues in a geriatric population, because that field is changing rapidly. And there's some pretty bad data and there's some pretty bad practices out there that are probably harmful. And so making sure that you're talking with your PCP about that is really important. But different drugs do different things.
Your excellent talk has reminded me of something that I couldn't understand when I first experienced it, and now I do. I was visiting a colleague at the University of Alaska Fairbanks who was studying hibernation. And the study has the Arctic ground squirrels, which go into really deep, and I was holding one, it's like it's frozen. But during the deep hibernation, in the middle of the hibernation, the Arctic ground squirrels warm up to normal body temperatures, which can cost an enormous amount of energy, and then they fall asleep. And they got up there and they went to sleep for a while, and then they slept for a while, and then they went back down. And so they paid an enormous energetic price to wake up just to sleep. And I think I now understand that much better than I did when I watched it.
Yeah. So the interesting thing that might very well. So one of the things that we see in our sleep is we undergo cycles. So when you go to sleep, a healthy night of sleep is usually made up of 3 to 5 sleep cycles, right? And the question is, why is that? Mice don't really do that. Mice oscillate between non-REM sleep and REM sleep, non-REM sleep and REM sleep, and they're just kind of jumping back and forth the whole time. But if you look at human sleep, humans go into slow wave sleep and they hang out there for a while. And then they go up to REM sleep and they hang out there for a minute. And then they go back down to slow wave sleep and they hang out for a while. And then they go back to REM and they hang out there for a little longer. And so the question is, why is human sleep stages so consolidated? Why so long? And why does it have to oscillate? And one of it might be that, you know how in a bucket, if I have a bucket of water and I just go like this really fast, all I get is turbid motion. I get motion that goes everywhere. But if I slowly go like this, what happens in that bucket of water? I start a whirlpool. And so it could be that those long consolidated sleep phases that humans have is needed for entraining fluid movement through a giant human brain versus a mouse brain, which is just like, and so the obvious test of that is sperm whales, right? So we get a sperm whale, we put the sperm whale in an MRI, and then we measure this. No, that's not really true. But wouldn't that be such a cool study? Maybe the NIH, maybe the National Science Foundation will fund the sperm whale MRI study.
I have a nightly process of waking at probably 3 to 4 hours, 5, after going to sleep, getting up because I have to go to the bathroom. I stumble. I imagine a lot of people in this room may have the same problems that I do. Stumble into the bathroom and within a couple of minutes, few minutes, come back to bed and go back to sleep. And this is a regular pattern. It's a nightly pattern at this point. Is this the kind of thing that would be considered sleep deprivation?
So first, I'm sure your experience is entirely unique in this room.
Yeah, I'm sure that's true.
Certainly, yeah. I don't experience that.
You're not old enough.
So I think the question that I don't think it is regarded as sleep deprivation. I think the question is, are you able to initiate sleep again? Yeah. Right? And if you can, that's great. Yeah. And then if you add your periods of reasonable sleep in between, do they add up to what you need such that the next morning you're not feeling like you got hit by a bus and you can think properly, right? And if the answer to that is all yes, that's not sleep deprivation. That's just how your body works, right?
Thank you. That's encouraging.
Well, both of my questions have been responded to: meditation and the sleep break. Meditation and the sleep break for going to the bathroom. So I'd just like to compliment you on your. I'd like to compliment you on your allusion to the sperm whale in the MRI. That just boggles the mind.
I'll let you all know that maybe that's in 2 years, depending on how the federal funding situation goes. I'll let you know if they let us do that. Interestingly, sorry, we're going to go off on a tangent here. Did you know that dolphins and sharks sleep with half their brain at a time? Dolphins and sharks, their brains sleep a hemisphere at a time. And some migrating birds do, yeah, which is super fascinating. And so I have a colleague. She did work in my lab, and then she got her own. She's a professor now at Medical College of Georgia in Augusta. And once in a while, dolphins and whales wash up on the shore in North Carolina and in Georgia, and they take the brains. And so she started it's kind of a random curiosity, but she started doing these neuropathological things that we do in people. She started doing them in whales and stuff, because it'd be interesting to find out if things hemispherically actually look different in the kinds of animals that have that kind of flipped, that kind of biology. So it's kind of crazy. Sorry.
Yeah, this is sort of a philosophical question, but do you think that the lack of ability to rapidly translate from the rodent studies to the human studies in the long length of time has to do with the fact that mostly when we study rodents, we take a mouse off the shelf, and when we study humans, and our study of the rodent model is like studying one human. And when we study humans, we have great genetic diversity.
Yeah. So the question is, why are rodents such terrible models? And why are humans also such terrible models? I think genetic diversity is obviously a big part of it, but that's a two-edged sword, right? So if I want to study, so the issue is in our mouse studies, our mice, they're called most of the time, they're called inbred strains, which means that the mice are bred brother to sister for the last 50 years, which means that if I've got 1,000 mice, it's like they're genetically identical twins, right? And so if I see an effect, guess what that effect is not being driven by? Genes, right? So if I'm interested in non-genetic Alzheimer's disease risk factors, it's actually great, because when I study a human population, if I'm going to do my next study in this group, so when I bring this drug to a clinical trial, maybe some of you will volunteer. The bad news is you guys are so different from each other. I have to study so many of you. The good news is if I find an effect, it means it's an effect that is valid for a diverse population. So I need a bigger group to manage that diversity, right? But the benefit is if I do get something that's relevant to a diverse population, I think there's pluses and minuses. To me, the mouse issue is actually, it's bigger than that. It's that I'm talking about transport, right? So if I wanted to say, hey, and I want to talk about how hard it is to move a tractor trailer full of teddy bears, right? And I want to understand what influences the rate of transport. Is the rate of transport I care about between Seattle and Marysville? Or is it between Seattle and Massachusetts, right? Totally different factors on those two different scales. A mouse brain is this big. A human brain it's about this big, right? Mouse sleep, different than human sleep. Humans, most of their brain is white matter, which is axons, the fibers. Mice hardly have any white matter at all. Hardly any. Mouse astrocytes, they're very simple, right? They have 5 branches. Human astrocytes, which are the support cells of the brain, have 150 branches, right? So there's huge interspecies differences that we have to deal with. One solution to that might be non-human primate work, but non-human primate work is really tough. It's expensive. There's issues having to do with just sort of the ethics of it, right? So there's lots of problems. We are doing a non-human primate study right now down in Oregon where we're studying a drug that enhances this process in monkeys. And one of the reasons we're doing that is there's questions that we need to be able to answer in a big primate brain that I can't answer in a person, right? And so that's tough. So we use all kinds of models.
In our household, we both have a family history of Alzheimer's disease. So for about 6 years ago, we learned about the glymphatic system and how it can clear the brain of amyloid and tau. And we're told at the time that because of that, it was a good idea to give the longest possible fast at night between the last meal in one day and the first meal in the next. I wondered if that makes sense in the face of the most recent research.
It's so interesting. First, have you read? There was a series of articles that said if you sleep on your side, your brain will clear itself better. Has anyone read that? It's a thing. Do you know the study that the data that supports that? It came out in 2013. Guess what? Guess what kind of animal it was in? Rats. And rats. So, what's the difference between a rat and a human? Well, some humans are rats. Some rats are human-like. So what's the difference between a rat and a human? Anything that matters? We're upright. Rats are this way. Think that matters? Rats are nocturnal. We're diurnal. Right? So it's tricky. Is it true that how you sleep will affect this? Maybe. It might be. No, we don't have the data. So what you're talking about is fasting. It's hard to tell. With the fasting thing, there's sort of two different plays there. One is a metabolic play having to do with calorie restriction, right? And which probably relates to cardiovascular health. If I improve my cardiovascular health, will I improve this process in humans? Do we have any data for that? We have no data for that. Do I think it's probably true? Yes. Are you going to do yourself harm if you improve your cardiovascular health? No, you probably should. You should do that today. So maybe there's a metabolic play, but there's no actual data for it. Part of it may be circadian. There is a circadian influence to this process. Guess what that was shown in? Mice. Is it true in humans? We don't know. So if I have a better circadian rhythm in a human, do I get a better process? Probably. Do we have any data for that? No. Would it be better if my circadian, which is my 24-hour cycle, aligned better with my normal sleep-wake cycle? Yes. Is glymphatic function the reason for that? Maybe. Are there other reasons that are more supported by data? Yes. Should I have a good circadian system? Yes, do that. Right? But we don't have the data. So that's an example of we read all sorts of things, but very frequently it's either based on a very small amount of data that isn't really related, or it might not even be related to real data at all.
We have time for one final question.
Okay, maybe you covered this in the previous talk. What is the difference between I'm trying to think of the glymphatic and cerebral spinal fluid?
So the question is, what is the difference between the glymphatic and the cerebral spinal fluid? And it's even more confusing than that. Are you ready? What is the difference between the lymphatic, and the cerebral spinal fluid? I didn't tell you about that because I didn't want to lose everyone. So in 2012, we made up the word glymphatic. That word it's a new word made up of two pieces. Glial, support cells of the brain dependent on glial cells, and lymphatic. One of the things that the lymphatic system does is it clears out waste between the cells. So it's a glial system serving a lymphatic function. So we called it the glial lymphatic or glymphatic system. Okay. 2012. 2015, lo and behold, someone actually found lymphatic vessels around the brain. So I gave a TED talk in 2014, and the thing I said at the very beginning was the brain doesn't have lymphatic vessels. Within a year, I was proven wrong. It does. We just didn't know they were there. But they're not in the brain. They're out here. So there's lymphatic vessels embedded in the meninges around the brain. The real lymphatic vessels. The glymphatic system is how things exchange from the brain out to the fluid. So you can imagine clearance as a two-step process. Step one, glymphatic clearance to the CSF. Step two, movement from the CSF into the lymphatic drainage. 2 steps. What is the CSF? The CSF is the thing that's being pumped by the glymphatic system. It is also the intermediate space. It's the holding space. Stuff gets cleared to the CSF, and then it gets taken up out of the CSF to be dumped into the blood. Okay? Lymphatic, CSF.
We're going to have to wind it up here. Let me tell you that there are two articles on reserve in the Let's Talk Science part of the library. One has to do with describing the very research that you just heard about. The other one has to do with the importance of sleep. So if you want to use look at those articles, please read them in the library and leave them there for other people to read. This has been an incredible talk. We're going to have you back next year to bring us up to date on all right. Thank you very much. Jeffery Iliff.